MES Marampally e-learning.
Hello everyone, welcome you all to my session. I am Dr. E.Sc Antony, Assistant Professor, Department of Biosciences, MES College Marampally. The topic of my presentation is antigen processing and presentation, and for this session the targeted audience include students from all streams of life sciences at both undergraduate as well as postgraduate levels, and all those who are interested in knowing about the intriguing facts about our immune system. Today, the world is facing a pandemic due to novel coronavirus termed COVID-19, and we are going through trying times that we have never ever experienced before. In this current scenario, the much talked about topics include how to boost our immune system, which are the different means to eliminate this particular virus, which are the different mechanisms by which our immune system fight against these pathogenic organisms, etc. That means the most important factor that concerns us is our health and nothing else. In this context, let us discuss some of the important aspects of our immune system. Our immune system fights to guard our body from all invading disease-causing pathogens, and in immunology we use the general term antigens to describe such foreign agents. These antigens are foreign agents which have to be eliminated from our body, and our immune system works very hard to eliminate them. The important components of our immune system include cells of the immune system, then organs of the immune system, various innate barriers such as skin, and the various chemicals produced by the cells of our immune system such as antibodies, then cytokines, etc. Cells are actually can be described as various of our immune system, and the important cells involved in immune response include B cells, T cells, macrophages, other white blood cells, etc. And among these cells, T cells are an important subset, and they are associated with cell mediated immunity. Cell mediated immunity is the branch of immunity that deals with intracellular pathogens, then cancer cells, etc. Now, but for the T cells to respond, there are some important prerequisites. That is nothing but antigen processing and presentation. And in this topic, in this session, we will be dealing with the antigen processing and presentation. With this brief introduction, let us move on to the details. Before going to the process in detail, let us understand some of the important terminologies. First one is antigen processing. It is the process by which the protein antigens from pathogens are degraded into peptides. Next, antigen presentation. It is the process by which the degraded peptides associate with the cleft of MHC molecules in the cell interior. They are then transported to cell membrane, and on the cell membrane they are displayed to TCR. For your kind information, these MHC molecules, these are nothing but molecules which are associated with self non-self recognition of a body, and these TCR, they are T cell receptors for antigen recognition. Next is MHC restriction of T cells, self MHC restriction of T cells. It is the absolute requirement of T cells for the presence of MHC molecules to present the antigenic peptide. That is, if a T cell has to recognize the antigen, this antigen has to be presented on the surface of this MHC molecule. And these MHC molecules, they are of two types, MHC class 1 and class 2. There are two important subsets of T cells also, CD4 plus T cells and CD8 plus T cells. The CD4 plus T cells require antigenic peptide to be presented on the surface of MHC class 2 molecule, whereas CD8 plus T cells require antigenic peptide to be presented on the surface of MHC class 1 molecule. Next, we have to be familiarized with antigen presenting cells. Antigen presenting cells, these are the actual cells which perform the processing and presentation of antigen. There are two different types of antigen presenting cells. One is professional and the other type non-professional. Professional antigen presenting cells include dendritic cells, macrophages and B cells. Non-professional APC include fibroblasts, glial cells, pancreatic beta cells, thymic epithelial cells, thyroid epithelial cells, vascular endothelial cells, etc. There are two important pathways associated with antigen processing, exogenous pathway and endogenous pathway. Exogenous pathway happens if the antigen is present outside the antigen presenting cell, and the antigen has to be internalized or engulfed by processes called phagocytosis or endocytosis, and it is processed inside these vesicles or compartments called endosome. It is processed and it is presented on the surface of class 1 MHC molecule. In case of the other pathway, that is endogenous pathway, the antigenic peptide or the antigen, it is present inside the antigen presenting cell and it is processed inside and it is presented on the surface of class 2 MHC molecule. Such antigens include tumor antigens, then intracellular pathogens such as bacteria, viruses, etc. Now let us discuss the first pathway, that is processing of endogenous antigens by cytosolic pathway. In cytosolic pathway, the antigens that have to be degraded, they are first bound with or they are first tagged with a protein called ubiquitin. So, this process requires utilization of ATP, and the formed ubiquitin protein conjugate, this is degraded inside a multifunctional protease complex called proteasome. So, this proteasome, it is actually a cylindrical particle consisting of 4 rings of protein subunits with a central channel of diameter 10 to 50 angstrom. This proteasome cleavage, it also requires ATP. And in the right diagram we can see, so in the left diagram peptide fragments are generated from proteasome. In the right diagram B, we can see the peptides generated from proteasome, they are now present in the cytosol, and from the cytosolic compartment it has to enter the rough endoplasmic reticulum, and this transport, it takes place to a transporter protein, and that transporter protein, it is called transporter associated with antigen processing or TAP, and this process it also requires ATP. And now after entering the rough endoplasmic reticulum, these peptides have to associate with MHC class 1 molecule. The MHC class 1 molecule is present on the membrane of rough endoplasmic reticulum. It is produced by polyribosomes located along the membrane of rough endoplasmic reticulum. And in the diagram you can see certain proteins called molecular chaperones associated with this MHC class 1 molecule. So, here we can see calnexin, then calreticulin, tapacin. These are the 3 molecular chaperones, or they are nothing but they are mentors. They are like mentors and they assist the proper folding of the class 1 MHC molecule, and it will also ensure that proper binding of antigenic peptide will happen on the antigen binding cleft of class 1 MHC molecule. Now, let us see the assembly of peptide with class 1 MHC molecule. So, the molecular chaperones are involved. The first molecular chaperone that associate is calnexin. So, before that let me briefly tell the structure of class 1 MHC molecule. It actually contains two chains, two polypeptide chains. One is alpha chain and the other chain is beta 2 microglobulin. So, first alpha chain is involved. So, this molecular chaperone calnexin will bind with class 1 MHC molecules alpha chain, and next it will, so thus it will help the proper folding of alpha chain, and next beta 2 microglobulin will come and bind to alpha chain. As this happens, the calnexin it is released, and now the other remaining two molecular chaperones tapacin and calreticulin will associate with class 1 MHC molecule, and next the next event that takes place is the peptide should bind to the antigen binding cleft. So, this binding of antigenic peptide, it is assisted by tapacin. So, this tapacin, it is present on the rough endoplasmic reticulum membrane, and it is, and this calnexin it is replaced, and now the other remaining two molecular chaperones, that is tapacin and calreticulin, will associate with class 1 MHC molecule. This tapacin expanded form is TAP associated protein. So, its function is to bring the TAP transporter, which actually transport the antigenic peptide into the proximity of the class 1 MHC molecules. So, the purpose is now the class 1 MHC molecule can easily acquire the transported antigenic peptide. And now the MHC molecule has the antigenic peptide in its antigen binding cleft, and through Golgi complex it will be transported to the cell membrane and it will be displayed on the surface of the cell membrane. Now, let us see the second pathway, that is processing of exogenous antigens, and that is by endocytic pathway. So, here the antigens are present outside the antigen presenting cell, and in the left side the left diagram shows how these exogenous antigens they are taken inside the antigen presenting cell or how they are engulfed. These exogenous antigens they are internalized or engulfed by processes such as phagocytosis, endocytosis, or by both processes. The internalized antigen later will move through increasingly acidic compartments. These compartments are called endosomes, and the acidity increases progressively from pH 6 to late endosomes or endolysosome, pH 5 to 6, then finally lysosomes which has still acidic pH 4.5 to 5. These endosomes also contain lots of hydrolytic enzymes such as proteases, nucleases, glycosidases, lipases, phospholipases, etc., which along with the lower or acidic pH will help to degrade the antigenic peptides into oligopeptides of about 13 to 18 residues. Now, so we have the peptides degraded now. And next what happens is simultaneously class 2 MHC molecule will be produced by polyribosomes located along the membrane of the rough endoplasmic reticulum. And when this class 2 MHC molecule is synthesized by the polyribosomes, it will be associated with a protein called invariant chain. So, the invariant chain it will be bound to the antigen binding cleft of the class 2 MHC molecule. And the purpose of this binding is to prevent any endogenously derived peptides from binding to the cleft of the class 2 MHC molecule, because in this compartment, that is in this rough endoplasmic reticulum itself, the processing, some parts of the processing of the exogenous and the endogenous antigens also take place. And now this class 2 MHC molecule invariant chain complex, which is produced on the rough endoplasmic reticulum, it will be transported through Golgi complex, again through endocytic pathway, and as this complex moves through the endocytic compartments in the similar way as the antigenic peptide also travelled, the enzymes will cleave the invariant chain, and the total peptide is not cleaved off. A small peptide, a short fragment is left as such in the antigen binding cleft, and that short peptide or fragment, it is called clip. Clip stands for class 2 associated invariant chain peptide. And now what happens is, next this clip has to be replaced by the actual antigenic peptide. So this replacing or this exchange is brought about by non-classical class 2 MHC molecule, it is called HLA-DM. So this HLA-DM will now exchange the clip with the actual antigenic peptide. In this slide, we can see a comparison between the cytosolic as well as endocytic pathways. In the left side, we can see endogenous pathway, towards right side exogenous pathway is given. In endogenous pathway, we can see the antigen is endogenous in nature. It is degraded by the proteasome multifunctional enzyme complex. The peptides generated transported to the endoplasmic reticulum via tap transporter, and inside the rough endoplasmic reticulum there is the class 1 MHC molecule produced. With the help of molecular chaperones, proper assembly of antigenic peptide takes place with class 1 MHC molecule. Then it moves to Golgi complex. Then it is transported to the cell membrane surface and it is displayed on the cell membrane surface with antigenic peptide in its binding cleft. Whereas in endocytic pathways, we can see first the class 2 MHC molecule will be produced by polyribosomes located along the membrane of the rough endoplasmic reticulum. Then we can see an invariant peptide chain is also associated with, or it is bound in the antigen binding cleft. Later it will move through the endocytic compartments, and in the process the invariant chain is degraded except leaving a small short fragment in the binding cleft called clip. Later simultaneously the external antigen, exogenous antigen, it is internalized by phagocytosis or endocytosis. It is degraded in the endocytic compartments or endosomes, and the degraded peptides will replace or exchange the clip protein, and this exchange, it is mediated by non-classical MHC molecule called HLA-DM, and now it will be transported to the cell membrane surface and it is displayed on the cell membrane surface with antigenic peptide on its surface. That is all about antigen processing and presentation. Hope you enjoyed the session. Thank you for patient watching.